Hello, and welcome back to Honors Biology. So today what we're going to talk about is mitosis and regulation of the cell cycle. So we talked about, chromosome, how chromosomes are made and replicated, and we've gone through the stages of interphase, G1, S, and G2. We're going to let cells spend most of their life in this interphase in one of these three stages. Now let's talk about what happens when cells are actually ready to divide. So we've gone through G2 and we're now entering that M phase of this. So we're first going to talk about mitosis. Mitosis happens in all somatic cells. So all body cells, this is the process that happens. This is how the cells actually divide and the nucleus splits apart. So really we're focusing on the nucleus here. How does the nucleus and DNA divide? There's 5 major stages to mitosis. The acronym we remember for it is PMAT plus C. So, because there's 5, so prophase, metaphase, anaphase, telophase, then plus cytokinesis happens at the end there. So that's what we're going to look at all these 5 stages. So let's dive into it. So in prophase, what happens is the nuclear envelope, so the envelope surrounding the nucleus disappears. The chromosomes condense, and we can actually now see the sister chromatids and the centromere. And then we also get spindle fibers starting to form. So we get 2 new structures that help us during mitosis. We get these things called centrioles. So the centrioles are these like tube-looking things. These are centrioles. And then what's forming in the middle here, these are the spindle fibers. So you're able to see what's happening here in prophase, right? The chromosomes be condensed. We can see them now just through a simple light microscope. We can see the chromosomes. We can see the centromere, so that middle part where the sister chromatids connect. This nuclear envelope is disappearing, right? It's fading away. And then the centrioles move to opposite sides of the cell with the spindle fiber starting to form. That's prophase. This is what prophase looks like. Our next phase is metaphase. Metaphase is probably the easiest phase to identify just looking at a picture. Because what happens in metaphase, all of the chromosomes line up in the center of the cell. I remember it as being middle, so they line up in the middle of the cell. All the chromosomes do. And what's happening also is that the centrioles have moved to the opposite sides. They move to the poles of the cell. And the spindle fibers that are forming actually connect. They connect to the centromere. So they connect to the middle part of each chromosome. So that's what happens in metaphase. The chromosomes line up in the middle. The centrioles move apart to opposite sides. And then the spindle fibers connect to the chromosomes. Now we're ready for our next phase, anaphase. What happens in anaphase is the chromosomes get pulled apart. So the sister chromatids separate into their individual chromosomes. So they get pulled apart. I always remember as anaphase starts with A, so there's a part, they're pulling apart. So, and they'll keep moving until they're in their separate sides of the cell. Right? So that's all anaphase is. The individual chromosomes, they get pulled apart by the centrioles and the spindle fibers. That's why the spindle fibers have to connect to the centromere. With that connection, then they pull them apart to the individual sides of the cell. That's anaphase. Our next stage is telophase. So telophase, it's the 4th phase of mitosis. And what happens is that chromosomes, they gather at their opposite ends, and the nuclear envelope starts to reform. So the nuclear envelope starts to reform, around our chromosomes now. That's what telophase is. And then the last phase of mitosis is cytokinesis. So cytokinesis, we actually get the cytoplasm pinches in half, right? We can kind of see it start happening here, how it's pinching in. We call this a cleavage furrow. A cleavage furrow starts to form here, and then cytokinesis, we actually pinch the cytoplasm in half. In the end, we get two new daughter cells. Now, each daughter cell, it's genetically identical to the parent cell, right? Because they replicated the DNA, and each cell now has a copy of the same DNA that the parent cell had. So they're identical. So that's your phases of mitosis, right? PMAT plus C, prophase, metaphase, anaphase, telophase, all right? Make sure you're able to go back and forth. Make sure you can tell me what stage a cell is in by a description, by me telling you what's happening, or with a picture. Okay? So make sure you're able to identify both ways of what stage a cell's in. Now let's just briefly talk about control of the cell cycle. Right? Like how do cells know like how fast or slow do they know to go through this process? Well, there's checkpoints at the cell. There's proteins that we call cyclins that control the cell at checkpoints, and there's three checkpoints. There's the G1 checkpoint, and so at the end of the G1 phase, we reach a checkpoint. Okay, and that the G1 checkpoint really all it's doing is just deciding, hey, we're gonna divide or we're gonna wait. That's all it's gonna do. We're gonna divide now or we're gonna wait. So the G1 checkpoint, if the cell passes that, it then enters S phase, and then it goes through S phase. Then at the end of S phase, we enter another checkpoint. The S checkpoint determines if DNA has been properly replicated, right? Because making sure that we get DNA replication accurate and we don't have, mutations that could cause problems, that's very important. So that's what the S checkpoint does is, hey, we replicated DNA. Is it accurate? Is it correct? Once the cell passes through the S checkpoint, then it goes through G2, enters into the M phase. We start with prophase, then we go into metaphase. Metaphase is where we get the mitotic spindle checkpoint, because this ensures that chromosomes are aligned at the mitotic plate or the middle. It makes sure that the chromosomes are lined up in the middle of the cell. Because if they're not lined up in the middle of the cell and the spindle fibers haven't attached to the centromere, then anaphase isn't going to work well. We're not going to get them to pull apart well. And then telophase isn't going to work well. And cytokinesis. So in the end, we're going to get two cells, yeah, but they're not going to be identical to each other. So what do these checkpoints, look like inside the cell? Well, here's our cell cycle picture. So here's where they're at. G1 is down here, or the G1 checkpoint, the cell growth checkpoint is down here, right? Occurs are the end of growth phase 1. Checks where the cell is big enough and has made the proteins for synthesis. And then if not, the cells go back through a resting period. We call it G0. Cells are at rest until ready to divide. Once it passes the G1 checkpoint, it then moves through, replicates DNA. At the end of replicating DNA, at the end of this S phase here, they go through the S checkpoint, or the DNA synthesis checkpoint. All right, and when it's occurring, it makes place. It checks whether DNA has been replicated correctly, and if so, we move on to mitosis. We go through G2, we start mitosis, and then if all said is done there, then we actually, fully divide and we get our two daughter cells. So this is how the process works and how it regulates itself. Now, cancer is if this process doesn't work, if these checkpoints don't work the way they're supposed to go. Cancer, what it is, it results of uncontrolled cell division. There's a mutation that happens that makes our cyclins, our protein checkpoints, not work. Right, the cells lose the ability to regulate their cell cycle. So, and then they just reproduce very rapidly. And these masses, or what form, that's a tumor. So typically, normal cell division, what happens is this, right? Normal cells grow and divide, they grow and divide. If their cell damaged, the cell is damaged in cell way, the cell will the cell will go through cell suicide or apoptosis. In cancer cells, though, we have a mutation that causes the cells to lose the ability to regulate the cell cycle. So then it just grows and grows and grows until we get there's just mass of cells formed. And that's not good because that can cause tons of problems. So that's it for mitosis and the regulation of the cell cycle. If you have any questions, please let me know. Other than that, I'll see you in class. Bye.